Thank you for coming to this third of the Van Horn lectures for 2009. And those of you who have been at the first two lectures, I would like to point out that there are a couple of things that did not go said or not mentioned, but which are really should be pointed out. For example, you can't just put lithium and iron and phosphorus in an aqueous solution at room temperature and expect lithium iron phosphate to happen. Even if you do, or let alone when you restrict yourself to conditions that are compatible with viruses. And it's true that you have 1 billion viruses who are helping sort out the work for you, but I'm sure there's a lot of sophisticated aqueous inorganic chemistry that's going on that Angela has not been telling us. But as somebody whose work has depended a lot on aqueous inorganic chemistry, I appreciate that there's quite a bit of tailoring involved. So that impressed me. The second thing that impressed me is not so much scientific as it is sort of the human side of science. And that's for someone who has not had any formal background in, say, electrochemistry, to decide that she has an idea for making a new way to make batteries. Anybody who's tried to break into an established field of technology realizes that there's a lot of inertia, there's going to be a lot of suspicion towards new ideas, and it takes a lot of courage. It takes a lot of confidence in the quality of your ideas to do the kind of work that Angela has done, not just in batteries, but in photovoltaics, in fuel cells, in electrochromic materials. And so that, I think, is also an impressive lesson that we're all going to be able to take from this series of lectures. And so with today's third lecture, we're going to learn about Angela Belcher's contributions and her group's contributions to materials for biomedical applications. So once again, let's welcome Professor Angela Belcher.
Thank you. Well, thanks for coming back for the last lecture one more time. Today I'm going to focus on what our efforts look like in terms of using biology, using materials for medical applications. Although because I back this up a little bit because this was supposed to be another one of the Darwin lectures, and because I had promised to put in our Sapphire work before and hadn't gotten to it, I'm going to tell you a couple of stories today. I'm going to tell you a story of yeast instead of a story of viruses, and how you can exploit yeast to make materials for you, and what you can learn from that, and how you can apply that to Sapphire, a very beloved material here in this department. Then I'm going to tell you a story about how we're using some of the same ideas on viruses and nanowires that I've talked about over the last couple of days to apply it to MR contrast agents for cancer diagnostics and cancer treatment. And then the last small story I'm going to tell you is one of instrument design, new ways of using atomic force microscopy for real-time studies of measuring antibiotics and their effect on cells in real time. And so those are the stories that I'm going to tell you today. But to start off a little bit, I wanted to tell you a little bit about our friend the yeast. These are the same yeast that are used to make beer and make bread. But we've been focusing on yeast to make materials instead. And this is in collaboration with Professor Dane Wittrup, who's pioneered yeast antibody display technology. He's at MIT. He's a colleague of mine. And one of the things that we like about yeast, that yeast are really big. They're about 4 microns in size. And so you can see them very easily in the light microscope. I didn't really go into the details over the last couple of days for when you're doing 1 billion different experiments using viruses, how you actually see the viruses when you're doing the experiment, because that's really hard. And so we looked at using a yeast technology as a robust technology of a way of making materials. Now, the downside of yeast is that they're spherical. A lot of things that we're interested in are anisotropic structures for wires. But let's talk about the good things about yeast, because there's a lot of really good things about these. Also, I should say these are easily transformable to undergraduate teaching labs because you do your screening with a light microscope instead of a TEM or something like that. So yeast are eukaryotic organisms. That's another advantage of these organisms, is that you can do the genetics in real time in a yeast. remember the viruses that I talked to you about. The viruses, you can make genetic modifications in them. In order to see your genetic modifications, you infect them into a bacterial host and use the bacterial host to make the modifications. So it's kind of one step removed. In a yeast, you can actually do this in real time. Yeast have DNA. There's some proteins involved in yeast mating. They're called AGA1, AGA2. These are proteins on the surface of the yeast. There's hundreds of thousands of these proteins on the surface of the yeast that you can exploit. Now, I'm not going to quiz you guys today, but remember how last time I said you could make DNA modifications in your virus, make modifications in the coat of your virus. Now you can make modifications in the genes of your yeast, and now you can express proteins or antibodies or really large proteins on the surface of your yeast instead. And one of the things I didn't tell you about in terms of the downfall of the technology of viruses is that you can only express pretty short peptides. Usually we're expressing things on the order of 12 amino acids or something like that. In a yeast, you can express whole enzymes, you can express antibodies, so that's kind of an advantage. But another thing that I like about them is they're nutritionally regulated. So they're living organisms. And you can feed a yeast glucose, or you can feed a yeast galactose, another sugar, and you can turn on different genes, and you can say, feed it glucose, it doesn't express the protein of interest, and you can feed it galactose, and now all of a sudden it turns it on, and now it can make semiconductors. And then one of the things that I like about that, I like this idea of a material stem cell, if you will. Stem cell is not a bad word anymore. That what if you had a yeast that had all the genes to be any kind of material you want it to be, you just fed it the right sugar, it turned on those genes, and it can make those materials. And so that's something you could do in real time, and so that's appealing to us. Now you can display, thousands to hundreds of thousands of these molecules on the surface, nutritionally inducible. Feed them one thing. They're not adherent to surfaces in general, and they're huge. They're 4 microns. This is a technology for materials we worked out in my group. Eric Crawlin was a student in my lab. You grow them up in these flasks in the lab, and now you're going to get them to start doing what you want them to do through the same kind of evolutionary process we talked about before. Have a billion different yeasts that are all exactly the same, but they're expressing different proteins on the surface. Force them to interact with the material of interest. Wash the ones that aren't off. And now at this stage, when your yeast of interest is stuck to your material, you don't have to go in with an acid and remove it and things like that you had to do with the viruses, because you guys probably know that yeast bud. And so now it'll just bud its progeny off. And now you can just collect its progeny from solution. And only the progeny from the parents that stuck to the surface you can collect, and you can go ahead and use that for materials. So there's a lot of advantages about them. Here's our screening process. Take a library, interact it with the material, wash them off, grow it off, go through this process just like we showed before. This is on single crystal cad sulfide wafer. These are non-expressing yeast that don't stick. And now you look at the light microscope, all these yeast are happily sticking to cad sulfide. So our idea for this was wow, yeast, we don't have to think small. We can think big. We don't have to think about short peptides. Let's think about entire antibodies, because antibodies is how it works in our body in terms of binding and selecting antigens, right? And so we put human antibodies on the surface of yeast as a library, and we made our yeast with our human antibodies on them bind to a bunch of different materials. And we were going to pull out these wonderful antibody structures that antibodies selected for cad sulfide. Antibodies don't like materials. Antibodies have a lot of structure to them. You're dealing with a nice single crystal flat material. They don't stick. They don't like it. When we went and we sequenced all this, what we found, here's some sequences, is that almost every single time we got actually early stop sequences, truncated antibodies, short antibodies. Short antibodies are peptides. Peptides is which work well with viruses. And so instead of saying this is a failure, we said, oh, this is great. What this tells us is we were right all along. Peptides are the right choice for materials. And then my students did very, very clever things. These are some really good, kind of hardcore bioengineers. they decided to give random link sequences to a yeast. Let a yeast pick any distance, any size it wants to pick for a protein randomly. And let the yeast decide what length is the best for materials. And I thought that was very, very clever. And so they looked at all the data, and yeast like between about 12 and about 20 amino acids. So we were lucky. We were right in the right ballpark the whole time. Yeast like these materials like these non-structured short amino acid sequences to give you the best binding and the best highest specificity. So it was a failed experiment in some ways, but we learned a lot from it, and we built new libraries based on this. I've never built a library in my life. My students built new libraries based on this. And so they went and they found a couple of yeast, and here's a cute little yeast that's budding. It's making its progeny there. This is cad sulfide in solution. And then they did a combinatorial screen to look for yeast that could grow semiconductors. And these are cad sulfide nanoparticles. They're going to be fluorescent. So now you can just look at them under a UV lamp, and the brighter they are, the better they are at making cad sulfide. And now you could go in there and collect those out, and now you have a sequence for some of these kind of materials. And these are the nice little cad sulfide nanoparticles that our yeast made. And if you go and you look at these sequences, here's a sequence. This is our best cad sulfide grower. And you go and so histidines, remember that's that magic amino acid that if you ever get stuck, say histidine, because it can be neutral or positive at physiological pH. It's a very important amino acid. Look at all these charged histidines in this group here. And so my students went and did what's called an alanine scan. Alanine is kind of a boring amino acid. It has a methyl group on it. So it's kind of boring in the fact that it doesn't even think about it as being very reactive. And they went in all these places here, 1, 3, 4, 5, 7, 8. They did a single amino acid substitution, pulled out histidine, and put in something more boring like alanine. And then they asked how this could bind. And so this is wild type here. And so this is actually percent coverage, how many yeast are covered on a surface. So the higher it is, the more yeast are covering it, the better it's binding. Does that make sense to you guys? OK. And so this is our this is actually changing the first amino acid, aspartic acid, to alanine. And it actually doesn't change it much or makes it actually a little better. But we found is taking any of these histidines out, any single of those histidines out, decreased its activity. And so again, we said histidine's important, but every single one of those histidines is important in its placement for binding. And so then my students went, and I actually forgot exactly what this is. Oh, they looked at this sequence here versus 6 in a row versus 5 in a row, 4 in a row, 3 in a row. And so the interactive strength depends on the multiple actions of histidine residues. And so then I showed this a little bit before. So we looked at cad sulfide, zinc selenide, cad selenide, gold. See if we could come up with a rule for these. And the way that they did that is they took these yeast and they put a short peptide sequence on it, and they put all alanine, all histidine, all mono amino acids of all 20 possibilities. And I said the other day that histidine came out as being really important on all materials tested. Cysteine came out to be important, and that's not so surprising, okay? That all of these materials like histidine. But what was surprising was that mattered what was next to those individual amino acids. And I said that you can make a fingerprint, like all of these materials like binding histidine. But it's these amino acids that are next to it that tell how good it binds. And so these amino acids are upregulators of binding to gold, and these amino acids here are downregulators of binding to gold. And you can start making trends like secondary aromatics, positive charges, and glycine are up regulators, down regulators or hydrogen binders, negative amino acids, and hydrophobic for this kinds of material systems. And so my students went in and they designed from scratch something that could bind all three based on these rules, looking at up regulation, down regulator, one that would bind just gold, and one that would bind just zinc sulfide. So you can start to learn these kind of rules from doing it combinatorial first, seeing what you pull out, doing site-specific mutations, looking at patterns, and rational design. And that's what they were able to do. And I'm pretty proud of that. And so then they decided to look at this material, sapphire. And I don't remember why we started looking at sapphire. but I think it was that we could actually buy these different crystal faces, and it seemed very stable, and it was transparent. That was another reason that we looked at it. So my students actually ordered C axis A and R, and they decided to look at throwing a yeast library at these different sapphire materials. And what they found was that there was not a lot of specificity between the different crystal faces, that in all three, basic amino acids ruled. Naive means this is basically random or background. So all three of these like basic amino acids, arginine and lysine. Sorry, I'm tired. None of them really liked negative amino acids. And they were a little bit more neutral, but also didn't like the hydrophobic amino acids. They also found that the C face was the most permissive. It would bind most everything. But they found 7 clones within these sub-libraries of all these different possibility testing that seemed to be the winners. And then they also said there's no single obvious amino acid motif. Remember, some of the data I showed you is that we'd start with 1 billion and we'd fall out with 2 sequences, 2 out of 1 billion. Never saw a single sequence that fell out from solution as being specific for sapphire. So my students, again, really smart students, said, we're going to rationally design materials based on the principles we learn for sapphire. And they said, we know positive is important. We're going to take this amino acid lysine, has an amine group on it, and we're going to go glycine. Glycine, remember, has a hydrogen on it. Glycine, lysine, glycine, lysine, glycine. Then we're going to put them in groups of 2, and then we're going to put them in groups of 3. And we're going to test the affinity of the spacing of these lysines and glycines. And now we're going to look at arginine, another positively charged group, and we're going to do the same thing. And now this is percent coverage, so the higher the percent, the better it binds. And this is K1. It's the best binder. Putting them 2 together drops the affinity. Putting 3 together drops the affinity even more. And then lysine has a better affinity than arginine. And these are what the structures of these look like. But we had some pretty good binding constants, about 80 plus or minus 45s binding constant, nanomolar for these materials. That's actually not bad. But then they said, well, how important is structure? And so they took that same one, K1, that was that one. They also put one with a disulfide bond in it, made a loop, made a kink into it. And then they also put proline. Proline is a natural amino acid that adds kinks into things. And now they tested K1, and they found that actually adding the kink to this increased its ability to bind to these materials. So chain flexibility is important to orientation towards the charged surface. And then they did some molecular simulations of these different sequences on the R face of sapphire. And they found that this alternating glycine K actually increased the contact of these binding sites with the surface of the sapphire. And here's the 3 Ks in a row where there's less contact with the surface. And so Steve Kopman, a graduate student in my lab, did all the modeling and a lot of the experimentation on these. And I remember why we were doing this now. The reason that we were doing this is, and this relates to medicine, and everything after this I promise will be related to medicine, is how many people have heard of hist tags, histidine tags, a way of actually being able to have a protein that has a bunch of histidine on it that can bind to another substance through a nickel interaction. It's a way of making a bunch of proteins in a cell and purifying only the protein you want because it has a histidine residue on it. And people have always said to me over the years, it'd be great to have another tag besides histidine, because you can't put multiple histidine tags in a cell and then try to separate them out. And so we started thinking about this as a new kind of tag. And so a sapphire tag instead of a nickel tag. And so what my students did is, a big problem is when you have something like this that's displayed on the surface, if you now take it off to the surface, does it still work? Big problem, big issue. Pushed under the table up to this point. You guys have trusted me on this, that it'll still bind. And so we actually took a protein, maltose binding protein, expressed our tag this 2K1, this K1 tag on it, and showed that we could actually bind this to a surface and it was still active, and it actually bound pretty good. We studied some of the kinetics of this. And then we found if we actually made that sequence twice, and then we called 2K1, that we got binding constants that were about 1.3 nanomolar, which is a really tight binder. And so this could be used as a different kind of tag. The histidine is about 30 plus or minus 3 nanomolar binding. And so that's compared to the nickel NTA. And so this is just kind of the rationale. I mostly brought that up because I know there's Sapphire fans out there, and then wanted to throw something kind of controversial in the mix here. My next story is about genetically templating magnetic nanowires for targeted MRI contrast agents. And this is work done by Rana Ghosh, who is graduated here as an undergraduate in biomedical engineering, then went on to Rice, and he's a fabulous postdoc. Eugene Lee, who's a graduate student in my lab. Kim Kelly, some of this work was done when she was a junior professor at MGH. She's now at UVA. And it's all done in collaboration with my group. So the motivation here, need for early cancer detection. About 90% of the deaths occur due to metastasis. Look for nanoparticle probes for magnetic imaging. Nanoparticle clearance due to protein absorption is an issue, so to look at optimizing that. So look at filamentous structures have recently been shown to stay larger in circulation in vivo. And then work on active tumor targeting with antibodies against known markers. There's not nearly enough of these. Can we produce new antibodies or new kinds of markers for early diagnostics of certain kinds of cancer? And we're looking at prostate cancer, colon cancer, breast cancer. So again, we're going back from yeast to viruses, M13 bacteriophage, and we're going to use the fact that it's long and skinny as a wire for magnetic contrast agents, fluorescent agents, and then directed targeting towards cancer cells. So we're going to use the phage to do the selection just like we did before, but now for magnetic materials. And I showed you before how fun it is to grow semiconductors on the tip of a virus, but now we're going to think about growing them magnetic wires that on one length, so one end has a protein that can bind to a prostate cancer cell line, bring with it an MRI contrast agent, bring with it a fluorescent contrast agent, and then eventually bring with it a therapeutic. And what people normally use selection for is selection for proteins. And so we've been working on phage display, and this is not something that we've pioneered at all. There's wonderful groups around the world that are using phage display looking for tumor targeting agents. Now, we started looking at SPARC. SPARC is a protein overexpressed in many cancers. It's called secreted protein, acidic and rich in cysteine. And this was work by my colleagues Kim Kelly at MGH and Ralph Weisletter. And what they did using this regular phage display kinds of techniques, not regular, but stuff they perfected in their lab, is they found a clone actually that's very specific to bind to this SPARC, which is this protein that's overexpressed in cancer levels. And so we're going to use that as a basic mechanism for tagging cancer. And then we're going to use iron oxide Fe2O3 particles as our MRI contrast agent. In my group, we worked out a way of growing, this is Eugene Lee, who's an awesome synthetic inorganic chemist in my lab, worked out a way of growing iron oxide nanoparticles, functionalizing them with amine groups, and making them water stable, very small particles of many different sizes for months and months and months in water. And this is an improvement because a lot of times people will make these iron oxide nanoparticles and they won't be stable in water for a long time. And we're going to need these to be able to bind to our scaffold or whatever we're going to do for our MRI contrast agents. Now,
are those crystalline?
Yes, those are crystalline. Yeah, it's not, sorry. You could actually see it if you came up here. You know, we don't have nice TEMs like you guys. But yeah, it's, these are crystal. Yeah, this is actually crystal material, yes. Now you can take these and attach them to our M13 phage. They make these linear kinds of structures. But the question was, when you do this, and this is something we didn't know at the time, when you completely cover this, and we have better covered pictures. I'm not sure why we always use this picture. But when you completely cover this with the inorganic material, remember, you have an active region up here that's one minor coat protein, and an active region down here that's another minor coat protein. This is the part we want to be able to bind to our cancer cell. So we need to check to make sure that you're not covering that, and that you can indeed use that for cell targeting. And so my students did that. They did a typical assay that's called an ELISA, enzyme-linked immunosorbent assay, that showed that the functional peptide against its spark sequence could still bind just fine at the same time that it had this inorganic material, and that was a big relief to us because before that we weren't sure that it was. Now the materials that we're going to, the kinds of contrast we're going to do is we're going to do dark MR imaging, transverse relaxation, and in this kind of MR imaging, you don't get brighter, you get darker, okay? And so you have your nuclear spins that are not aligned, you have applied magnetic field, and you get a net magnetization M that's parallel. You apply RF pulse, and the magnetization is perpendicular to your magnetic field. Now you put in your iron oxide particles, and they're homogeneous in the field caused by this iron oxide. The nanoparticles cause phase decoherence, spin decay, magnetization becomes zero, you get loss of signal, it seems darker. And so in this case, when it gets darker, that's when you have your material that's being able to bind. And so now we did some relaxivity data on these phage-based nanoparticles, and we compared it against Clio, which is an already used MRI contrast agent with iron oxide that's basically a polymer-linked system that brings in iron oxide particles, and the relaxivity of this is about 30 to 40, and our nanoparticles, which are kind of large, 12 particles on the phage, are about 6. Our particles by themselves are about 65, and our magnetic phage are about 59. And so our magnetic phage have a higher relaxivity than the commercially available cross-linked iron oxide nanoparticles, and now we have to think about using those in targeting towards cells we're trying to identify. The kinds of cells that we used were called C42B, a prostate cancer cell line that has highly expressed SPARC, that sequence that I told you about. And here's how we did these experiments. We took our phage that had the iron oxide particles on them. They had the anti-SPARC sequence on one end, and the magnetic particles on this end. We actually labeled the phage at the same time with a fluorescent agent, so we could follow it fluorescently and magnetically at the same time. So this is the M13 nanoparticle negative control here, where you have M13 that has the fluorescent marker and has the magnetic nanoparticles, but doesn't express the anti-SPARC sequence. And you're looking at, sorry, there's no scale bar here. You're looking at, basically, you're not seeing very much fluorescence, so you're not seeing very much phage either binding to or being taken up by these images. Over here is the sequence that's, again, it's the 2-gene sequence, the 2-gene system I've been talking about for days. This one has the genes to be able to have the iron oxide particles and target the SPARC cells at the same time. And you can see that these are actually increased in these C42B prostate cancer cells. And now we actually wanted to look at the magnetic uptake by these cells. And now this is actually 1 over T2 over here versus iron concentration. So we're going to be looking at the total amount of iron that's taken up by these cells. The black dots are iron oxide standards. The red squares are iron uptake by our negative control, meaning no SPARC sequence. And the blue ones are our SPARC sequence iron oxide. And we get a 50% improved uptake of these targeted magnetic nanowires to these cells. You know, these are the kind of things you get that you get really excited about, but you need someone who works in this field to bring you back to reality often. And so now we're going to actually look at some different controls. And these controls are two. One is on the red are those prostate cancer cell lines that I told you about, that a highly expressed spark. And this one here also expresses spark, but it's not highly overexpressed like the prostate cancer cell line. And so now what you can look at over here, in this case, you're trying to make things darker. And so this is the one that actually expresses spark. This is the one that overexpresses spark. And so you're seeing a decrease in your MR contrast, which means that yes, some cells, spark is not specific to only cancer cells, but when it's enriched in a cancer cell, then you actually see more MR contrast. These are more experiments that the real biologists made us do, which are time uptake of these, which are how much do you take up after 30 minutes versus how much do you take up by 24 hours. And so you're getting more uptake of these cells as a function of time. So this is very beginning work. We're actually going to in vivo models for this inside rats. So there's a lot of things we need to do. Do different size nanoparticles, work on our targeting with the M13. We're looking at the in vivo models. Actually, Rana is the student from here, is actually going to UVA at the end of the month to test some of these in UV models. Looking at biodistribution with our nanowires versus other kinds of structures. And then actually start to put both fluorescent and therapeutics on these materials. So this is even newer work and even a little bit more far out. But I showed you those gold nanowires yesterday as, what are we looking for? Fuel cells, catalysis for fuel cells, and these gold-silver alloys. Now we're actually going to look at the synthesis of gold nanowires for near-IR biological imaging. This is work done again by Rana and Eugen. It's desirable for minimally invasive optical imaging for cancer diagnostics. This near-IR absorption, 700 to 1100, is ideal for in vivo biological imaging, known as the water window. There's been numerous agents that have been developed for NRI imaging, dyes, gold nanoshells, and gold nanorods. However, there's a problem with circulation times. And we think, and recently people have shown that more anisotropic objects can stay in circulation longer before they're cleared. And so we thought we would test some of these with our gold structure. So this is the sequence that I showed you before that grows gold, beautiful gold wires with 100% conversion, almost 100% conversion efficiency. You can actually look at the absorption spectrum of these made at different ways and over different times. And you can start to modulate this depending on synthesis. These are the wires as synthesized. You vortex them, they go back into solution. So these are very long structures. And I showed you before that we can make alloys, and alloys are going to be interesting for us in terms of tuning the near-IR spectrum, so the ratio of the gold to silver. These are some UV absorption of our alloy nanowires. Here, this is a gold to silver ratio of 4:1, a gold to silver ratio of 2:1, and a gold to silver ratio of 1:1. And you see these peak shifts here, but then you also see a rise in the near-IR regions as you increase the silver. So light absorption at 1100 nanometers increases with the amount of silver content, which is our near-IR response. And this is not very good data yet. This is just very new data showing some of what this looks like over this window. So we've been looking at these high aspect gold wires. We should be able to do the same things that we did with our targeting and imaging for our magnetic contrast agents, but now we're going to look at near-IR properties, and we're currently looking at uptake targeting and visualization of these gold and gold-silver nanowires in solution. Okay, my last mini talk, which is, again, it's involved around nano, is involved around bio, but has no viruses in it and no yeast. This is work on real-time dynamics of antimicrobial peptides on the nanoscale using high-speed atomic force microscopy on single cells. This is one of my postdocs, Georg, who invented the instrument that we're going to talk about. It says it's a prescription for one of those new super antibiotics. You won't just get better, you'll get even. And this is a collaboration by, the instrument design is all done by Georg in my group, and then other people as well, and I'll tell you about this today. So basically, my postdoc built this instrument in our lab as a way of looking at, it's basically video speed atomic force microscopy in solution. And so we were looking for a problem. I wanted to watch viruses be born, and that ends up to be a little bit hard. But we started looking at bacteria. And there's a natural host defense peptides, possibly a next generation antibiotics, small peptides extracted from frogs and insects, and they're a part of their natural defense system. They act on bacteria, some viruses, fungus, and some cancer cells. And they have a potential for broadband antibiotics for therapies. These AMPs are thought to are very positively charged proteins, short peptides, and they're thought to break open cells by this carpeting of the outer leaf of the peptides on the surface, integration into the lipid bilayer, making holes, and then transporting inside and basically blowing them apart. A lot of work on this. So we're going to use AFM to look at this. And everyone here knows about AFM. And AFM has a very sharp tip. It's a way of looking at surfaces. You have a laser bounce off, go to a photodiode here. It can also be done to look at as you heard last year, probably in the Van Horn lecture, of molecular interactions as well. And you can look at it for conducting surfaces, electrochemical potential, all kinds of things. But it's slow. It's too slow to measure dynamics. And there's about 1 scan line per second, or about 8.32 minutes per image. And so it's mostly looking at stationary samples. And so this is AFM circa 1987. This is 2008. And there's been a lot of improvements. This is a high-speed, small cantilever AFM. You have optical lever detection, MEMS cantilevers, AFM in liquids, and some things called small cantilevers. So small cantilevers can be used as probes for biophysics. There's certain improvements: high resonant frequencies at lower spring constants, lower Q factors, higher force resolution, and faster detection. And Georg published these before he came to my lab as part of his PhD thesis in Paul Hansma's group. Now, this is the video speed AFM that they developed, which has 8 frames per second, 512 pixels per line, 256 lines per frame, and a maximum scan range of about 15 microns. So this is atomic force microscope image that you can use like a scanning electron microscope and move it around at video speed. So Georg came and built this in my lab. And so we decided to look at a short peptide, a lysine-rich peptide, 15 amino acids. You can see all those lysine groups in there. And its proposed action is membrane destabilization of a bacterial cell. And so these are some images, a 1 micron scan line here. And we're going to look at a real-time video of this antimicrobial peptide acting on an E. coli bacteria. And there's about 7 seconds per image. And this is tapping mode phage imaged in liquid. And there's some other details if you're interested. Here's 2 happy E. coli. And this looks really easy, the fact that we can that we could get our E. coli, stick to a surface, add liquids, and then image this at video speed. I said, guys, just go out and do that, figure out how to do that. It's actually a lot harder. There's a lot of technique into getting these bacteria happy on a surface in solution so that you can image them at video speed. And it took us 6 months or so, it took my students 6 months or so to work this out. Now, there's going to be lines that come across, and that's when we're adding the antimicrobial agent to this. Right there, add it, and what you can see within 7 seconds that the morphology of this bacteria starts to change. Remember that atomic force microscopy, you're going to be looking at the surface, visualizing the surface at the nanoscale of what's happening. I'll play this a couple of more times. It's smooth. You add it, it becomes wrinkled. And you can actually even see little holes start to develop within just a couple of seconds, way less than a minute. You can actually start to see holes happen in real time in this bacteria. Let's see, how do I get out of this? Let's see. Okay. Now, one of the things that's really fascinating to us, and again, we're not experts in infectious disease, and my husband always thought I didn't have good ideas in infectious disease. He's an ID doctor, but then he thought this was kind of interesting. Now, here's 2 E. coli right next to each other in a very, very small space. We're adding quite a bit of antimicrobial peptide into here in a small space, and the mixing is probably pretty fast. But the rate at which these respond, or the rate at which these end up dying, is actually pretty different. And that's really interesting from a genetics point of view, why two E. coli that are right next to each other are going to respond to the same, from the same colonies, respond to the antimicrobial agent differently at different times. And so this red one here actually responds very, very quickly. This is seconds after injection, and you can see the blue curve here. And now we go and we measure the roughness, surface roughness, just like you always do in AFM. And now we're going to correlate surface roughness with cell death. And here's a movie of this happening. Each one of these frames is 24 seconds per image. Here you're adding the antimicrobial agent, and you look at different roughnesses of these bacteria in the same field of view as a function of time. But we wanted to actually prove they were dying, and so we did my student then built a combination fast AFM fluorescent bioscope under my assistance. No, actually, he did it all himself. And so here's a regular AFM image here. Now we're going to do something called live dead staining, where you give it a stain where green is live and red is dead. It has to do with how permeable that cell is to the stain. And here's just normal E. coli, no antimicrobial agent. And here it is, normal happy E. coli. There's some dead, no antimicrobial agent. Here's where they start getting rough after they're given the antimicrobial peptide, and showing that they're all dead at a function of time. And you can kind of watch the individual ones die by the roughness and correlate that with the fluorescence image. And here's let me just go back one, the concentration. This is about twice the concentration, the minimal inhibitory concentration, which is about 12 micrograms per milliliter. Then we actually just did some regular AFM where we took these antimicrobial agents, put magnetic particles on them, and we're kind of looking at their distribution around the top of these E. coli, showing that they're actually sticking to the surface of these E. coli. And we don't have, this is just very, very beginning data here. And now we did an experiment, which I don't have the data here to show you about, with a very talented professor at MIT in biological engineering, Scott Manalis. You might have seen he had the Nature paper of the year in 2006 or 2007. I don't remember what it was. But he was also an undergraduate in Paul Hansman group like I was. We were there at the same time. And he's an AFM expert. He invented, I can't explain it this well, but he invented an inverted cantilever where you could actually flow liquid through the cantilever, and you could measure very small changes of mass in liquid. And he had a big Nature paper on measuring the mass of a single bacteria cell. So we started working with him, taking a single bacteria cell, putting in our antimicrobial agent, and we saw right away that in the presence of this antimicrobial agent, our cell lost 30% of its mass. Okay? And we thought that was interesting. My students went through the calculations, and they said, well, it has to be more than just water. It has to be losing more than just water. And so we did some experiments where we took these antimicrobial, so we took these E. coli, we put in these antimicrobial agents, and we're assuming that they made holes in them, like we saw that they did in the AFM, punch holes in them, and then we're going to collect the supernatant of these, filter it through a 10 kD filter, and look at if there's any proteins by just a regular protein gel. Look at any proteins coming out to see if these are losing proteins and how fast they're losing protein. And this is a silver-stained protein gel. It's I think a trisglycine 10 to 20% protein gel. This is our ladder. It's really, really overstained. We had to overdevelop it because there was not a lot of protein coming out. This is not treated. Actually, they're washed cells, but not treated with our peptide. And then the same cells washed and then treated with a peptide. And this is actually looking at a trace of that, which is showing that we're getting a population of proteins out after treated with the antimicrobial peptide out between this 80 kilodaltons and what is that, maybe 40 kilodaltons. So we have a population of proteins that are being released from these cells very rapidly as the action of these. Now these antimicrobial peptides are really, really small. They're less than 10 kD, and those get washed away. And so we think this is interesting. We haven't seen that people have shown this before. We haven't seen that people have watched antimicrobial peptides and starting to look at the mechanism, watch it how they work in real time. A lot of times people look at what happens after they're dead. And then I'll actually start to try to make some meaningful mass changes in these as they're dying. And then when they're dying, what are they spitting out? And that's kind of where we are with that. So that ends my 3 lectures. And I'll be happy to take questions on this. And I thank you for all your attention and your all your sharing of your ideas and experiments over the last several days. I really, really enjoyed it. I set up some collaborations and hope to get some good papers out of those in the next year or so. So thank you for your attention.
Okay, here we go. All right, questions? Jim.
So, in the last series of studies you showed that the bacteria die, you talked about 2 things there. One was that 2 bacteria very close to one another died at different rates. So I'm thinking that's related to where they are in the cell cycle.
It's definitely a possibility. Although we did look at some that were actually reproducing during the time, and whether they were reproducing or not reproducing, I didn't always seem to make a difference. But if you have any ideas of how to clock cell cycle in a controllable way, I'd be really interested in that.
The other question I have that comes out, you start off with a beautiful genetic selection with
Yep.
yeast. Could you do the same with bacteria to understand the mechanism? In other words, which ones don't die? Can you put a selective pressure with these peptides over time and come out with the bacteria that have mutations, even if you have to facilitate
Yep.
those mutations and then look at roughly, etc.?
Yeah, that's a great idea, and we're trying to figure out a little bit. So I hadn't thought of it in terms of stepped all the way back and done it as a bulk system. Then you're getting into, I'm good at viral genetics, easy. I'm not good at bacterial genetics, a lot harder. So it gets at more complications. But my students are starting to look at that. Can you start to identify? But the thing is also that they're not really, sorry, I'm really tired. They're not really resistant to the antimicrobials because they eventually all die, but they definitely slow down their resistance, slow down their death, and so they have delayed response. And so I think that is a big question. I think that's the big check mark I would be able to do is to try to understand why. And I'm going to think about your idea of backing it out and doing more of a genetic selection. It doesn't click in my head quite yet, but I'm tired and I haven't thought about it. Thank you.
So I just had a question for clarification with the magnetic nanoparticles on the viruses. Were those the contrast enhancing agents themselves?
Can't remember, actually.
Yeah.
I mean, did we use the iron oxide we used in the MRI contrast agents for the outside of the bacteria?
Yeah, I was trying to remember.
I think we did actually. I can't remember.
All right.
I mean, I actually maybe never known. Yeah.
Because there were at least two things that you attached
Yeah.
to those.
Yeah.
There was one that would glom onto the cancer cell.
Right. Yeah, yeah. So that was just a short peptide. That was taken to AMP and just through EDC chemistry, coupling the magnetic particle on it. It didn't have to be magnetic. It could have just been a 2,6 semiconductor particle. It could have been anything because we're just looking at size.
Okay, but so you were using that as a way to zero in on the cancer cells by making
Yeah.
them more visible.
Yeah.
The iron oxide, was it, do you happen to know whether it was cubic or hexagonal iron oxide?
You know, I have to go back. I have to go back and look for sure. I'm sure I have that on my computer, but
But your quarter is cubic.
yeah.
I think it's the Mayan stuff at our ops lab, right?
Yeah. Well, no, we actually made this is a different synthesis method, but I have to go back and look and see. I think it's cubic. It's cubic.
Just had a general question about the nucleation of these nanoparticles. So you have a peptide on the outside of a virus, and you have some atoms that are coming out of solution, for example, like the nickel or cobalt, whatever it might be. How exactly do you end up with an actual nanocrystal? I mean, I can imagine that you'd have maybe a couple of atoms that would attach themselves to the outside of a peptide.
Yeah.
But how do you end up with an actual crystal? Does it act as a nucleation seed?
Yeah, we think it does act as a nucleation seed, and there's a whole bunch of experiments I didn't tell you about, which is if your peptide binds too tightly, it won't nucleate. Those are a series of experiments that we did that we never take our tightest binders. Our tightest binders never correlate with our best nucleators. And we've mapped that out. And we're usually in the micromolar binding constant regime that we're getting the best nucleators of materials, because if they bind too tightly, it really doesn't allow for other atoms to come in, and it works much more like a poison. The exact mechanism of this nucleation, you know, we've nucleated over 40 kinds of materials now. We just don't know the answer for.
So as an alternative, could you use those viruses that act more like a poison to be a filtration mechanism?
Yeah. We look at that for lots of different kinds of functions. One is cleaning up toxins in the environment, or trying to get rid of one kind of material versus another. Yeah.
Have you found any findings, for example, like lead or?
Lead's easy. Yeah, we were working with arsenic. Actually, the hard part about that was, I'm trying to remember, was that getting the right arsenic species, bind to enrich for the right arsenic species in solution, kinds of things we looked at.
Well, there's,
Of course, you've talked a lot about mineralizing stuff. And I want to go back to your very first slide.
First day?
Tuesday.
Okay.
You
Tuesday.
talked about abalone.
Yeah.
And as you know, we both worked for a good long time on molluscan shells. And I concluded that we knew damn all about how biomineralization really works in terms of something that's really quite fundamental. I wonder if you would comment as your work trying to direct mineralization, give me some insight into really the way
Abalone
works.
No, I can't give you the insight to how abalone work. And abalone was too complicated, and so that's why we went to something much simpler. Because as you remember, in abalone, at the time that I was isolating peptides from abalone shells, soluble peptides from abalone shells, there were many different species, some of which were very anionic, some of which were very positive, and they worked in concert. And that was too complicated for me, and so I went to try to simplify it out more, to look for one particular amino acid sequence, one short peptide, because those are long peptides also. And remember, they're also glycosylated. They're post-translationally modified, and that made it a lot more difficult. I was worried that the post-translational modifications were going to be crucial for mineralization, and we're not going to have those kinds of post-translational modifications in these virus bacterial systems. And whew, that wasn't true. In our case, the amino acid sequence was important. The spacing between the amino acids were important. As I said before, there are upregulators and downregulators of amino acids next to things that are nucleators. My students have started working. I have a student that's right now that's very ambitious, is working on modeling a model for how proteins mineralize. And we'll see what comes out of that in the next year. So I've gone back to calcium carbonate in the last year as part of our CO2 flue gas to building supplies program, where we're getting organisms to convert CO2 to HCO3 and then HCO3 to different kinds of carbonates to use as building supplies. And we're going back and looking at the rates. We're doing some kinetics. There's never really any kinetics done on mineralization in calcium carbonate from solution. And we're going back and we're doing that for sequences and chunks of amino acid sequences, looking at the kinetics for mineralization of different sequences in real time. So if you know kinetics that we're missing, but it's also much more complicated because we're breaking things down by shorter amino acid sequences to try to pull out some of those fundamentals.
I was always impressed at how unsatisfied I found these synthetic experiments trying to do biomineralization
Yeah.
on the kind of insight that you'd like to have on how organisms create these wonderful structures.
Yeah.
And that's a very creative concept, too, to consider CO2 not just as a waste to sequester, but as a useful raw material.
Yeah.
Put it back to use. There's one last bit of information, distinction. We've already mentioned that Professor Belcher is our first female Van Horn lecturer, our youngest Van Horn lecturer, our first MacArthur Fellow Van Horn lecturer. And just before today's lecture, she found out yet another distinction. She'll be our first Van Horn lecturer to be singled out by Rolling Stone magazine. They've just come up with a list of 100 people who have changed America, and you are included on that list as number
31, yeah.
Number 31 with 100 people. So that's also our first Van Horn lecturer who qualifies as a rock star.
Well, thank you. I had a wonderful time. I appreciate your hospitality and teaching me so much about things I didn't know before I came. So I appreciate that.
Well, thank you. One more round of applause.